Hello, you guys. We're going to continue on in the Unit 2.1 notes. In these notes, in this video, we're going to look at Topic 2. In Topic 2, we're going to talk a lot about those membrane-bound organelles that are found in eukaryotic cells. Those membrane-bound organelles form this system of internal membranes that we call the endomembrane system, and a lot of them are coordinating between one another and kind of working together and doing things with each other. And so we're going to kind of walk through that. And so here's a little picture kind of a lot of the structures we're going to talk about in this topic. This is the endomembrane system, which again is this internal system of membranes found in eukaryotic cells that form these membrane-bound organelles that are coordinating a lot of tasks with one another. And you'll see a lot of things moving between these different things. And so there's the organelles that make up the endomembrane system include the nucleus, the endoplasmic reticulum, which we call the ER for short, the Golgi apparatus, and then there's these things called lysosomes that we'll talk about, and a variety of other types of vesicles and vacuoles, and the plasma membrane that surrounds the cell. And so let's start with the nucleus. That's an easy one. That is the place where the DNA is stored inside of eukaryotic cells. And that DNA, like I said, is organized into chromosomes. So chromosomes are single pieces of DNA that can be very, very, very long. In eukaryotic cells, we have linear chromosomes inside the nucleus. In prokaryotic cells, remember, usually they only have one chromosome and it's circular. And so the DNA that we find in eukaryotic cells, there's a lot of DNA that has to fit inside the cell, specifically inside the nucleus of the cell. And that DNA, what we find in eukaryotes, that DNA, that molecule DNA is wrapped around these proteins. And so with the help of these proteins that it gets wrapped around, it forms this really tightly packaged, very organized, condensed structure called chromatin. So chromatin is DNA wrapped around these proteins. That helps to really, really condense these chromosomes, these long pieces of DNA, so that they fit inside the nucleus. And so, like I said, humans, I said this in the last video, humans have 46 chromosomes. So you would have 46 of these super long pieces of DNA wrapped around these proteins inside each of the nuclei, inside each nucleus of every cell in your body. So anyway, that's the nucleus. Now, oh wait, the nucleus, why is this not in here? Okay, sorry about that. I just realized I accidentally deleted a slide, and then I found the slide and put it back on the notes. And I'm too lazy to restart this video, so let's continue on. So the nucleus, like I was saying, this is where the DNA is stored. The nucleus is surrounded by a membrane. It's actually a double membrane. So there's two sets of phospholipid bilayers that surround the nucleus. So there's a phospholipid bilayer surrounding the nucleus, and then a second phospholipid bilayer surrounding the nucleus. So we say that it has a double membrane. Within that that structure, the double membrane that surrounds the nucleus, we call that the nuclear envelope. So the nuclear envelope is those two sets of membranes that surround the nucleus. Embedded in the nuclear envelope, this membrane, or these set of membranes around the nucleus, are these pores, these openings, which are made of these protein structures that allow things to go into and out of the nucleus. And then inside the nucleus, you'll see oftentimes a very condensed region of DNA, or a very concentrated region of DNA. We call that the nucleolus. So the nucleolus is this region inside the nucleus where there's a high concentration of DNA. And this is actually where ribosomes are being built. And so ribosomes are those tiny little structures that build proteins. Those are actually made in the nucleolus region of the nucleus. And then those ribosomes are going to be sent out of the nucleus to go into the cytoplasm of the cell to build proteins. And you can see that here, they're actually going through these nuclear pores to get out of the nucleus. So the next structure then, if we kind of go from the nucleus outward, is the endoplasmic reticulum. This is a set of membranes that's actually connected to the nucleus, as you can see in this picture up here and here. And so this is a huge network of folded up layered membranes. inside the cell, which helps to increase the surface area inside the cell. So this provides a lot more surfaces for things to happen. A lot of times on these surfaces, there's chemical reactions happening, or we'll see in a second that there's proteins being built on this membrane. And so that's good, having more surface inside the cell for things to happen. And then there's two types of ER that you're going to see. There's going to be what's called the rough ER and the smooth ER. The rough ER has all these little dots embedded in it, and those dots are ribosomes. These are ribosomes, the things that build proteins. And the smooth ER is this piece of the endoplasmic reticulum, this set of membranes that has no ribosomes embedded in it. So let's start with the rough ER. The rough ER, its job, this is where a lot of proteins are going to be made, because this is where a bunch of ribosomes are. So there's all these ribosomes. Here's one here, super zoomed in on their ER. This ribosome is embedded on the rough ER, and that's where proteins are being made. So you can see this guy is building a protein. These amino acids are getting put together, and it's going to fold up into a protein. This is happening inside the rough ER. And then what happens to those proteins is they pinch a piece of the membrane of the rough ER, pinches off into what's called a transport vesicle that's now holding on to that protein. So you can see the protein is being made by the ribosome, and it's being built inside the rough ER, or put together here, and that's going to fold into it's going to start folding into its structure. And then as it does that, this a piece of the rough ER membrane is going to pinch off and carry this guy somewhere else, which we'll see in a moment where this guy is headed, carrying this protein. So that's the rough ER, and then these proteins that are being made by the rough ER, there's one of three main things that are going to happen, and this is something you guys do need to memorize. So these proteins that are made by the ribosomes and the rough ER, there's basically three types of proteins being put together here that are ultimately going to end up being one of these three types of proteins, or doing one of these three types of things. One of the things that might happen to these proteins being made by the rough ER is that they sometimes will be secreted by the cell. So in cells, a lot of times there's certain cells inside of organisms, including you guys, that secrete a lot of proteins, meaning like those cells release proteins out of the cell. So in this picture here, you can kind of see that happening. Here's some proteins inside of this vesicle, and it fuses with the membrane, and it's going to release those proteins out of the cell. We call that secretion. It's going to secrete those proteins. A good example is like your stomach cells, the cells that line your stomach. They're constantly releasing enzymes into your stomach they're secreting enzymes into your stomach, so those enzymes can break down your food that you're eating. And so that's an example. So those enzymes, those proteins, enzymes, were made by the rough ER. So any proteins being secreted by the cell, those were proteins who were put together in the rough ER. Another thing that happens to these proteins made by the rough ER is some of them become membrane proteins, or proteins embedded on the cell membrane. We'll talk about this more in the next set of notes, but there's a lot of different types of proteins you'll see on the cell membrane. Some of them are enzymes who are embedded on the cell membrane, catalyzing chemical reactions. Some of them are receptor proteins. We've kind of talked about these a little bit throughout the year so far, which bind to signaling molecules on the cell membrane and cause things to happen. Another thing that we've already talked about is that there's transport proteins that move things into and out of the cell. Anyway, all these proteins that are found on the cell membrane, these proteins were made by the rough ER, the ribosomes in the rough ER. And then lastly, some of those proteins made in the rough ER are going to become hydrolytic enzymes that are found in lysosomes. So this purple thing right here, this is a lysosome. We'll talk about that soon in this video. And inside the lysosome, there's these enzymes, which are proteins, whose job is to break down large molecules. And so they do hydrolysis reactions. So we call them hydrolytic enzymes. And we'll talk about these guys soon. But those proteins, these enzymes that are found inside lysosomes, those proteins were made by the rough ER. So those are the three types of proteins that are being made by the rough ER. So if we're talking about proteins being secreted from the cell, those were made in the rough ER. If we're talking about membrane proteins, any protein that's embedded on a cell membrane doing something, that's made by the rough ER. Are these enzymes found in lysosomes, those are made by the rough ER. Then we have, one thing I do want to point out before we move on is that not all ribosomes inside the cell are embedded on the endoplasmic reticulum, are embedded in the rough ER. So there are what we call bound ribosomes. Bound ribosomes are the ribosomes that are attached to the rough ER, building those proteins that I was just talking about. But then there's also, you're going to always see a bunch of free ribosomes. Free ribosomes are just ribosomes floating around in the cytoplasm of the cell. They're not bound to the rough ER. They also make proteins. The big difference here is that the proteins that are made by the free ribosomes, these guys that are not part of the rough ER, these ribosomes, those proteins are going to be the proteins that stay in the cytoplasm of the cell. So these are proteins that are doing their job inside the cytoplasm of the cell. In the jelly-like fluid, they're just floating around, they're doing reactions or have other jobs that they need to do inside of the cell. So they're not becoming one of these three. So basically, the other types of proteins being made inside of a cell who are just floating around the cytoplasm doing important jobs, those are made not by the rough ER, those are made by these free ribosomes just floating around in the cytoplasm. So that's the rough ER. And then we can talk about the smooth ER real quick. So that's the part of the ER that doesn't have these embedded ribosomes. The smooth ER is also where things are being built, but mainly in the smooth ER, the things that are being built are not proteins, it's actually lipids. There's a lot of lipids being made in the smooth ER. And so those lipids include like triglycerides, which are fats and oils, which store energy. Steroids, which are like your sex hormones, testosterone and estrogen, those are lipids. And phospholipids, which are the structures that actually build all these membranes, are found in all these membranes. Those are lipids. And so those lipids are made in the smooth ER. That's one of the functions of the smooth ER. And then beyond that, there is a variety of different jobs that the smooth ER has depending on the type of cell. One common example that depends on the type of cell. There's certain types of cells where the smooth ER actually has a really important role in detoxifying the cell. So there's chemical reactions that happen in the smooth ER that get rid of toxins inside the cell. A good example is your liver cells. In your guys' liver cells, they have a really extensive, large smooth ERs that are detoxifying substances that were found in your blood that these cells took in and now getting rid of. A good example is alcohol. And so when people are drinking a lot of alcohol, your body doesn't like that because the alcohol damages your cells. And so your liver and your liver cells are constantly taking all the alcohol out of your blood and the smooth ER's job is to take the alcohol and turn it into something not toxic. So that's the smooth ER. Then we can talk about the Golgi. So the Golgi actually works together with the ER a lot. So the Golgi is this series of flattened membrane discs that you can kind of see here that are called cisternae. These flattened, I don't even know what to call them, flattened disc membrane things here, systems of membranes flattened in series with one another. And so that's what the Golgi kind of looks like. You can kind of see in this picture up here where we're talking about in the cell. So this is not connected to the nucleus, but it's kind of, if we go from the nucleus to the ER and then further past the ER, you'll see the Golgi floating around somewhere. The Golgi has two sides or faces. There's going to be a side or a face that's facing the ER, and there's going to be a side of the Golgi that's facing the plasma membrane, the other side of the other side. And the side that's facing the ER is called the cis face, and the side that's facing the plasma membrane is the trans face. And so what happens in the Golgi is these vesicles that were pinching off of the ER. So in the ER, that's where lipids are being made and proteins are being made by the smooth and the rough ER. And a lot of times that stuff pinches off in these transport vesicles like I showed you earlier. Those transport vesicles then arrive at the cis face of the Golgi and fuse with the cis face of the Golgi. And then inside of the Golgi, what happens in all these little flattened discs here of membranes, there's a bunch of chemical reactions in here that modify those things that were initially made by the ER. So those proteins that were made by the rough ER, they're going to now be slightly modified. There's modifications that are going to be happening in the Golgi to kind of fine tune those proteins, help them fold correctly, and then be ready to now go operate and do what they need to do. And so beyond that, we're not going to get into specifics. There's a lot of specifics we get into about all these different types of modifications. Just know vaguely in the Golgi, it's modifying these packages that were received by the ER. So the ER put these things together, sending it to the Golgi. The Golgi is going to modify this stuff. And then the Golgi on the other side the transphase is going to then package it into a vesicle again and then send it off to its final destination inside the cell. So in the transphase, you'll see these vesicles pinching off, carrying these final products that have now been modified to their cellular destinations. And so that's what's happening in the Golgi. And so here's a good picture that kind of shows this flow of material arriving here from the ER, the cisphase, and then being shipped off on the transphase. Here's a good example of them being released from the cell. So like I said, a lot of times these proteins that were made by the rough ER, they're going to be sent to the Golgi. The Golgi is going to modify them, and then they're going to pinch off from the Golgi, and look what happens to them. They get released or secreted from the cell. So secreted proteins, they were made by the rough ER, but before they are done, they got to go to the Golgi to be modified and finalized. And so that's the series of events that is usually going to take place. So it's going to start from the ER, and then go to the Golgi, and then the Golgi is going to send it off somewhere. And so, like I said, though, just remember that these proteins made by the rough ER that are now being sent to the Golgi, and the Golgi, after they leave the Golgi, one of three things is going to happen to those proteins. They're either going to be secreted from the cell, like this picture, or they're going to become part of the cell membrane. So sometimes the Golgi is carrying proteins that are on the cell that are embedded in the membrane of the Golgi, and then it's going to fuse with the cell membrane and like go leave those proteins to be embedded on the cell membrane somewhere. That happens too. Or some of these become lysosomes. So some of these pinch off from the Golgi and now form lysosomes, which we'll talk about in a second, which stay inside the cell and break down molecules and stuff like that. So let's talk about lysosomes. So here's some quick information about lysosomes. Like I said, these are these vesicles that contain these special enzymes, these hydrolytic enzymes, who are really good at doing hydrolysis reactions. They break down these large macromolecules into smaller pieces. That's the function of lysosomes, is to break down this large material inside the cell that needs to be broken down. And inside the lysosome, it's actually very acidic, which is a pH that these hydrolytic enzymes function optimally at. In these lysosomes, they like to function at an acidic pH, and so in the lysosome there's a very acidic environment. And so these lysosomes, first of all, were made by the Golgi. So the Golgi received, so that, let's just follow the path here. There is these enzyme proteins, these hydrolytic enzyme proteins were made by the rough ER. They were sent to the Golgi. The Golgi modifies them and then packages them into a little vesicle that stays inside the cell and operates as a lysosome. So these guys were formed by the Golgi, but these enzymes inside ultimately came from the rough ER as they were going through the Golgi. And now they're doing their thing. So here's a common example is like the cell will sometimes engulf material from outside the cell. So here's some kind of large macromolecule from outside the cell. The cell's going to engulf it into one of these vesicles that you see here. It's called a food vacuole containing these large macromolecules. Could be a large carbohydrate or a large protein of some sort. And then these lysosomes will fuse with the food vacuole, and they'll release these hydrolytic enzymes to start breaking down that material into things that the cell can now use. So that's what lysosomes are doing a lot of times, is breaking down these large macromolecules back into monomers that the cell can then use those monomers to start building whatever it wants to build. And so that's a good example. This picture is also showing you a food vacuole that's being fused with a lysosome to then break down that material. Another common job of lysosomes, so they kind of have a variety of similar functions. They also can get rid of old organelles inside the cell. So if there's organelles that are no longer working inside the cell, like even something as big as like a mitochondria, it's just not working anymore, the powerhouse of the cell. And so lysosomes will fuse with those organelles and break down those organelles so that those components and those monomers can be reused to build other cellular structures. So a lot of times we compare them to like the recycling center of a cell. Like they're just constantly breaking down things back so that they can be reused in the cell. And then one last thing that lysosomes do is they also play an important role in apoptosis. We'll talk about apoptosis later this year, but apoptosis is when a cell programs itself to die. So it's called cell suicide. So this is when a cell decides, or someone tells it, like, you need to kill yourself, you're a bad cell. And then cells, they will do it. They will kill themselves, this is a good thing, actually. We want cells that are no longer needed or no longer doing what they're supposed to be doing. They need to go away. They need to die. And so this is a great process that happens where cells are triggered to just give up and to kill themselves. And lysosomes help with that process. That's all you need to know. And then let's talk about vacuoles. So vacuoles is kind of a vague term. It vaguely refers to any large vesicle that contains stuff inside of it. That's a vacuole. So it's a vesicle. It's a large vesicle. That's what a vacuole is. Like a food vacuole. That's a type of vacuole, and it's just a membrane sac that has something inside of it, or something's going on inside of it. Those are vacuoles. And there's a lot of different types of vacuoles that do a lot of different types of things. Like I said, food vacuoles, that's a common example where the cell engulfed something from outside the cell, and then it's going to be digested and broken down inside the cell. That's carried in a food vacuole. There's contractile vacuoles. I've seen this example come up on AP tests in the past, where these are vacuoles that are found in mainly in protists, so single-celled eukaryotes. So eukaryotic organisms that are single-celled, like amoeba and paramecium. These words you probably have heard of, but just certain types of organisms that are single-celled. And they have these special vacuoles, that these membrane sacs, that their job is to actually fill up with water inside the cell. So as the cell is starting to accumulate water, it fills up with water, these contractile vacuoles, and then it fuses with the cell membrane of the cell and squeezes. It can contract. Contract means it can squeeze, and it pushes out that water, which actually helps these guys move, these unicellular organisms to move. So that's something that we see in protists, these contractile vacuoles. It also helps to make sure that the cell doesn't fill up with too much water and explode. So sometimes these cells are in environments where there's just a bunch of water through osmosis going into the cell, and these cells need to get the water out before they explode and fill up with too much water. So those are contractile vacuoles. And then another example, I pointed this out at topic 1, is the large central vacuole in plants. This is a super big vacuole found in plant cells that takes up a huge majority of the space inside plant cells, that fills with water as well, along with other random things inside plant cells like waste material or nutrients or pigment molecules. It's like a little storage center for plant cells. But most importantly, it's filling up with a lot of water a lot of the times. And that's not found in animal cells, by the way, this large central vacuole, which is actually the technical term. It's called a large central vacuole found in plant cells. But anyway, those are some examples of vacuoles and different things that they might be doing inside the cell. And so this picture kind of summarizes this whole series of structures that are working in coordination with one another. And you guys can see there's this whole movement of membrane and material throughout the cell. So like these plasma membranes, which are made by the smoothie are, because they're phospholipids, there's just this huge movement, constant. It's very fluid, this flow of plasma membrane and cell membrane inside of eukaryotic cells that are maybe today part of the rough ER, but then that membrane moves in a transport vesicle and now becomes part of the Golgi the next day. And then those phospholipids, that membrane that makes up the Golgi, somehow ended up part of the membrane of the cell. And so there's this very complex, continuous movement of stuff going on inside of eukaryotic cells with lots of different things happening. And so hopefully, I don't know, that kind of makes sense in your head. The last thing on here is, and this is the last slide, but maybe the most important slide, is why in eukaryotes having this endomembrane system, this system of membranes, and these membrane-bound organelles, like why in eukaryotes is that good? And what advantage does it give them versus prokaryotes? So prokaryotes don't have all of the stuff that we just talked about. Everything that I just talked about in topic 2, none of that is found in prokaryotes. Make sure you know that. And so in eukaryotes, what advantage do they have then because of this over prokaryotic cells? And there's 2 main things that you need to know, just memorize. One of the things is by having this internal system of membranes and all these membrane-bound organelles, it hugely increases surface area, which there's just all these surfaces now inside the cell where things can happen. And for now, just kind of vaguely know that. This will make sense throughout first semester, but like on these membranes, there's a lot of reactions and a lot of things that need to happen on these membranes. And so in eukaryotic cells, there's a bunch of surfaces, a bunch of surface area for all this stuff to happen, which is helpful. And then the other thing is that it helps to compartmentalize, I'm saying that word correctly, it helps to localize these different types of reactions. So in the rough ER, there's certain reactions happening, building proteins. In the smooth ER, lipids are being put together or detoxifying the cell. In the Golgi, there's these different reactions modifying things. In lysosomes, there's reactions breaking down things. And so there's all these different reactions and different functions happening inside the cell, and they're localized into these different compartments that are surrounded by membranes, which increases the efficiency hugely of a eukaryotic cell. Whereas in prokaryotic cells, all this is happening, basically everything I said is still happening in prokaryotic cells, but not in these nice little organized structures. And so it's a lot more chaotic and messy and not as efficient in prokaryotes. So anyway, that's the end of topic 2. I'll see you guys later.